Hello. My name is Lenka Dohnalova, and it will be my great pleasure actually to be here today and to introduce my first PhD project that I've been working on here in Thaiss Lab at the University of Pennsylvania. And yeah, as Viviana suggested, it's actually gonna be very multifactorial research and it's very complex. So I will try to walk you through it step by step, and I hope that you can bear with me. And in case that you have any further questions, or there is something that you want to further discuss, you can definitely email me or get anyhow else in touch with me, and I will be happy to discuss with you the project further or more in depth. So what we usually don't think in the context of the exercise is actually all the factors that are being involved in can actually somehow influence our performance, and not only our performance, but also the differences between the individuals that we can see. The first group of factors are the genetic factors that not only in our paper, but also from many previously already published data sets that were made in the humans actually show that the genetic factors aren't particularly interesting, or at least important, in context of driving these huge inter-individual differences that we've seen. And it seems that they contribute way less than, let's say, 15 or 20% in the total variance in the exercise performance seen between individuals. So then the big question is if the non-genetic environmental factors, such as diet or our healthy or unhealthy habit, our age, our physiology, or our overall health status or microbiome actually can contribute to the exercise performance differences a little bit more than the genetic factors do. So then, of course, usually when we think about the exercise, we think about all the kind of superlatives that it is being linked to, and especially we know that the exercise is giving us a lot of benefits compared to the lifestyle that is more sedentary. And this was mostly studied in context of the cardiovascular diseases, or of course skeletal muscle physiology. And as we can imagine, for most individuals, it's very important also the metabolic benefits that are mostly mediated by adipose tissue, enteroendocrine system, and of course livers. But more recent studies actually show that the exercise might also be beneficial in terms of our immune system resilience, or increasing the life health span. And thus it's basically connected to the longevity. What actually I will be focused on today most, as I already suggested in the previous slide, is the non-genetic factor, and especially microbiome, that is basically residing our gut, and how it may actually interplay with the nervous system to affect our exercise performance. What we actually know is that the brain is very important receiver of, let's say, this beneficial outcomes of the exercise, not only due to the known cognitive decline that is being basically partially rescued by the exercise, but also we know that the entire nervous system is then flooded by the BDNF, noradrenaline, serotonin, but in context of this study is particularly important, the neurotransmitter dopamine that is being increased in very high concentration actually is present after the exercise. So why the dopamine is actually particularly important in the context of exercise? It's due to its basically ability to boost our motivation, but also this kind of pleasure-seeking behavior, or our reward pathways. So how and where actually the motivation is coming from? I will try to kind of simplify it as much as possible, but the motivation is being basically created or generated in the ventral tegmental area, which is part of the brain where the neurons are basically mostly synthesizing the dopamine. and then they are sending these dopaminergic projections towards the striatum, and they're particularly in the part that is called nucleus accumbens. And as you can imagine, to basically receive these projections, you need very high concentrations of the dopamine receptors being in the nucleus accumbens to be able to receive the dopamine from the VTA. Then the signal basically continues further towards the prefrontal motor cortex, from where it can basically, the signal can be passed back through the spinal cord to the muscles, and muscle can contract, and all the sensory signals can be then perceived. So first, before I actually start with the actual experiments and the project as it was done, I will first introduce the, let's say, the methods that we were using to assess the exercise performance in the mice. And we used two major models. One was forced exercise, where mice were running in the treadmills, and they're running on the belt, and the belt increases the speed, but also the inclination over the time. And if mouse either doesn't want or cannot anymore run, then it felt on this wired grid in the back. And 1 second presence on that grid is considered as one visit, and 50 total visits is then considered as basically that the mouse is exhausted, and that final time and distance is being recorded as time and distance to exhaustion run. And the second option is then to use the free voluntary exercise experimental setup, where each mouse was single-housed and had free access to the wheel, basically 24/7, and when it could run whenever it feels like. And there we were recording the spins every 1 minute throughout the day. So as I already kind of suggested before, we are interested mostly in the gut microbiome and how it can affect, if it can actually affect the exercise performance. So our first question, of course, then was if this is actually true and the microbiome itself actually can somehow contribute to the overall exercise differences. So in order to address this question, we basically use the C57 blacks wild-type mice from Jackson, and we divide them into two groups. First group was just regular control group, drinking regular animal facility water, while the second group received a broad-spectrum antibiotic mix consisting of neomycin, ampicillin, metronidazole, vancomycin, ciprofloxacin, and primaxine. And basically within approximately 2 weeks of drinking these antibiotics, the mouse has completely depleted the microbiome. And when we take these two groups and we run them in the both exercise and they get free access to the wheels, we actually see a huge suppression of the exercise capabilities, which it was like so extreme that we thought first it's just like due to some like either liver toxicity of these antibiotics or some like muscle physiology changes. But in order to exclude this, we basically did the same experiment, but with the germ-free mice setup, where the mouse never received any antibiotics, but still lacked the microbiome. And we basically saw exactly the same thing. We saw suppression of the exercise capabilities by approximately 50%, suggesting that the microbiome indeed is somehow involved in the exercise performance modulation. The problem of both of these setups is that basically it's very extreme. It's like either a full microbiome or no microbiome at all. So in order to kind of further narrow it down, we used each of these single antibiotics as a separate experimental group. And we again let mice drink these antibiotics for 2 weeks, and then we put them on the wheels and the treadmills. And what we saw is that there was this incremental decrease in exercise performance based on what antibiotics the mice were drinking for the extended period of time. And what we saw is that the neomycin in both cases performs similarly to control, while the ampicillin is a very poor performer and performs almost as poorly as the broad spectrum antibiotics mix mice. So then the question actually was, to further even narrow it down, which particular microbes or microbiome families might be responsible for driving this phenotype. So in order to address this question, we performed 16S sequencing from the stool of these mice before the exercise. And we were looking for the candidates that are basically very high in control and N-omycin and very low in the ampicillin or the completely depleted mice. And we found a couple of candidates such as Aristotelotrichaceae family, or also some members of Lechnospiraceae family that basically show this trend and were basically not present in the ampicillin depleted mice. So then we kind of were looking for particular species, and we just like kind of screened ATCC, which of these species, of course, are available that we can use for the monocolonization experiments. And what we found is that if we use the Eubacterium rectale or Coprococcus eutactus that were positively correlated with the exercise, and we monocolonize the gem-free mice with the species, they actually also increase the exercise performance almost all the way to the level to control SPF mice. While if we monocolonize gem-free mice with Blauschia producta that wasn't correlated to the exercise performance at all in either way, then we basically don't see any difference from the gem-free. While if we supplemented or monocolonized gem-free mouse with the Ruminococcus gnavus that was negatively correlated with exercise performance, we actually can even further suppress the exercise capabilities compared to like the gem-free mouse control, which actually really causally link the particular microbe that or group of microbes that are actually responsible for these exercise differences. But the problem usually is that it's not the microbe that does the thing. It's usually some product or some metabolite that these microbes can produce that are actually modulating the things that we've seen downstream. So in order to kind of like find which metabolite might be basically responsible for the phenotype that we've seen, we performed untargeted metabolomics from the single antibiotic experiment, and we took the cecum contents of these mice. And we actually here plot the heat map of the differentially abundant metabolites that were present in these mice. And what we actually see is exactly what we saw kind of in the exercise performance. The control was very similar to the no-mycin group, while the vancomycin and ampicillin that were very poor performers has very distinct and very different the metabolite composition compared to the good performers. And what then we basically just like went through the list of all these metabolites to find which group of metabolites, let's say, might be showing the same correlation and might be basically correlating with the exercise performance. And indeed, we find that the fatty acid amides, and here I'm showing just one example of olyl ethanolamine, is basically fitting the bill that it's is decreased with the exercise performance. And then basically the very logical question was if we basically give this metabolite back, if we can rescue this phenotype. So then we basically just get as many fatty acid amides that had similar colorations like olyl ethanolamine. We mix them into the diet, and we fed this diet into the control and antibiotic drinking mice for over a week. And then we put them on the wheels and the treadmills. And we actually see that just supplementation and giving back these metabolites into the system is basically enough to fully rescue the phenotype that was lost due to the absence of the microbiome in these mice. So then we kind of knew that there are these particular microbes that are responsible, and then there are also some metabolites that are linked to these exercise performance differences. But we kind of didn't really show that these metabolites are produced by these microbes that we suggest. So to address this, we did targeted metabolomics of the cecum contents again of the mice that received the Coprococcus eutactus that we saw that improved the exercise. And indeed, we actually saw that the levels were also higher which is kind of like very similar result of what we saw before, let's say with the single antibiotics and oleoethanolamine. Furthermore, we were actually very lucky that in almost similar time when we were performing these experiments, this paper that I'm citing here came out, basically showing that Eubacterium rectale and Coprococcus eutactus have this novel bacteria gene cluster that actually can synthesize fatty acid amides. So we were like, okay, great, let's basically take advantage of this knowledge and let's take these genes from Eubacterium rectale and clone them into the E. coli. E. coli is known to be not capable of producing these fatty acid amides. That's why it's also like good system or good bacteria to use for this cloning. So after we cloned the plasmid into the E. coli, we basically monoclonized germ-free mice with either the E. coli that had empty plasmid or the one that has these mutant genes. And we put them on the wheels and the treadmills, and we actually saw that to some extent, indeed, this E. coli with the mutate insert genes basically managed to rescue this phenotype, and they increased the running performance, which might have been due to basically increase of this oleoethanolamine in the cecum content that I'm basically showing you here in the ion chromatograph. So then we thought that really these particular metabolites that come from these particular microbes are responsible for this phenotype, but we actually don't know still what the phenotype is, right? We just see the difference, but we don't know where the difference comes from. So in order to kind of like very simplify this, I will just focus on the motivation pathway, and I will not basically say what all we have to exclude before we go to this. But it's basically, if this is the picture that I showed at the beginning, and in order to kind of like understand if indeed the motivation is affected by the microbiome presence or absence, we need to measure the dopamine concentration in these synapses. So the best way, or the very elegant way to address this, is to use the genetically encoded dopamine sensors. So the way this works is that you insert basically the optic fiber into the nucleus accumbens, and you measure the dopamine concentrations in live mice. And this in vivo imaging was done in both control and antibiotics mice post-exercise. And we actually saw that, as we kind of empirically know from humans, that right after the exercise, you have a very high spike of dopamine being released into the synapse. That's what makes us kind of like happy, and after the exercise happened. But we basically don't see this happening at all in the mice that are lacking the microbiome. And that was also further confirmed by ELISA from the nucleus accumbens, basically suggesting that these mice have very low motivational state, and they just don't want to do the exercise because it doesn't give them kind of like the pleasurable feeling. To further even more highlight the importance of this pathway, we did single nucleus RNA sequencing for the striatum before and after the exercise in both mice having intact microbiome and pleased microbiome. And we actually saw that indeed the stimulation of these neurons in the postsynaptic neuron is way lower in antibiotics mice compared to the control counterparts, which was furthermore than kind of like supported by qPCR from the striatum in these mice. So since we kind of like say that indeed there is less dopamine, and it seems that the downstream signal in the postsynaptic neurons is being basically blunted, then like can we basically modulate this pathway to basically increase our motivation or decrease our motivation? And it seems that that's really the case if we use sulpiride, which is antagonist of these receptors. Regardless basically how much more the control mouse has dopamine, the signal basically cannot pass to postsynaptic neurons, and these mice are demotivated and they just don't run. And they run as poorly as the antibiotics control. While you can imagine if we do quite the opposite and we stimulate these receptors, then regardless how little the antibiotics mice have dopamine, they basically get all of a sudden all excited about exercise, and they run basically as well as their control counterparts. So then then we basically kind of like support this idea that there is very little dopamine in the synapse in mice that lack the microbiome, and thus they cannot stimulate the postsynaptic neurons. So I think that like relatively logical thinking was, okay, it's just because there is not enough dopamine being synthesized in the presynaptic neurons, so then there is lack of stimulus into these synapses. So in order to address this experimentally, we did fiber photometry, where you basically insert the optical fiber into the VTA, and you use GCamp virus, which is basically encoding for the calcium release. And that's if you can basically say how much calcium was released after the stimulation of the neurons, you can use it as a kind of proxy of if and how the neurons are being stimulated in presynaptic neuron. And when we did this experimentally in control and antibiotics mice, we actually didn't see any difference, which means that there is basically in the presynaptic neuron, at least, there is no difference in the activation of these neurons. So there is nothing to suggest that at this level, there is basically lack of the dopamine synthesis, at least, because there is basically no input issue in these neurons if the mouse lacks or has full microbiome. So then we thought like if basically there is not problem with the synthesis, then there is probably a problem with the degradation pathway. So then we looked into the degradation pathway that is responsible for the decreasing the levels of the dopamine in the presynaptic level. And the enzyme that is responsible for this process is monoamino oxidase, encoded by MAO-A or MAO-B. And indeed, when we looked into the basically qPCR expression levels of this enzyme in the mice after the exercise, we actually saw that the mouse with intact microbiome has very low levels of this enzyme after the exercise, which basically suggests that they have way more dopamine because there is no enzyme that can degrade these dopamine boosts after the exercise. While this wasn't the case at all in the antibiotics mice, basically suggesting that this may explain the lack of dopamine in the synapse. So to kind of like experimentally address the MAO-A, we managed to basically inhibit its activity by using pargiline, which is antagonist of MAO-A and MAO-B. And after these antagonists been injected for a couple of days, and then we exercised the mice, we basically saw that this suppression of MAO-A activity was enough to increase the levels of the dopamine in the striatum of the antibiotics drinking mice, basically almost all the way to the level as the control mice that has full intact microbiome. And what was actually even more interesting is that this boost in the microbiome levels was enough to increase the exercise capabilities of these mice in both experimental setup in the wheels as well as in the treadmills, basically suggesting that if we manage to basically bypass this lack of the dopamine, we can increase our exercise capabilities. The problem is that we kind of now understand that these microbes and the products that they produce may suppress MAO-A and increase the dopamine, but we still don't really understand what is the pathway that basically brings this signal to from the periphery to the central nervous system. So there are, of course, many neurons that can basically be used to basically bring the signal up to the central nervous system. However, we focused mostly on the sensory nervous system. And as you can imagine, most of our organs, but also skin and also muscle, is innervated by the neurons that are then basically leading all their projections towards what we call dorsal root ganglia. And dorsal root ganglia are always in the pair for each vertebra, and so they basically innervate the entire spinal column all the way basically to the brainstem. And then when actually we sequence this single nucleus from these dorsal root ganglia, then we saw that actually, as it was kind of expected, that most of them are TRPV1 positive. What is TRPV1? TRPV1 is transient receptor protein vanilloid 1 that I think that is mostly being connected and known for its, let's say, ability to recognize and modulate pain. So it's mostly then heat or very high pressure, very low pH, or also, as highlighted here, it can recognize also molecules like capsaicin that are in hot spice, like red chili peppers. And all this basically stimulates these sensory neurons that can then send their projections through the dorsal root ganglia and spinal cord to the brain. So we basically took advantage of this system and we used the TRPV-DTA mouse model to specifically ablate all these neurons. So the way the system works is that any mouse that expresses CRE under TRPV promoter kill itself because it starts producing the diphtheria toxin subunit A. So when we use this mouse model, we actually saw that regardless of the microbiome presence or absence, these mice become very poor performers compared to their wild-type counterparts that are basically having intact sensory nervous system, suggesting that basically ablation of the sensory nervous system is enough to completely block the exercise kind of pleasure feeling. So you can imagine that we can do quite the opposite by using the capsaicin, which is the molecule I just said is agonist of these receptors. And if we use it in very, very small concentrations, then it can actually specifically increase the activity of these neurons. And when we inject capsaicin for a couple of days, we actually saw that in both settings, treadmill as well as the forced exercise, as the voluntary exercise, we basically saw a huge increase in exercise performance and basically full rescue of this phenotype just by stimulation of peripheral nervous system. And what was actually even more interesting is that this phenotype was basically really modulated by the suppression of the MAO-A in the central nervous system. And that, of course, resulted in higher levels of the dopamine in the synapse, which was basically comparable or even higher than the controls. And if we compare it to the basically TRPV-DTA mice, they had almost no dopamine, and that's why they also performed very poorly in exercise performances. So then actually what was even like more interesting is that when we were going through these data sets and we thought about the metabolites that we were using, we actually didn't really know if they necessarily bind to the TRPV1 because that wasn't published and we were not really sure if that's like true, and we want to further narrow it down. We look for basically other receptors that are being expressed by the TRPV neurons in the DRGs. And what we actually found, it was super interesting. We found that one of these receptors is actually cannabinoid receptor 1. And what's actually super interesting is that this receptor is actually binding all these molecules, all these fatty acid amides that we're actually using in the supplementation experiments before. So in order to basically address this, if we indeed need cannabinoid receptor 1, and in these sensory neurons, we basically kept injecting mice with the peripheral antagonist, which is AM6545, An advantage is that basically the entire sensory neurons are otherwise intact. Just this one particular receptor is being blocked. And when we did this experiment, we actually saw exactly the same as we saw in the TRPV DTA mice, that basically just suppressing these cannabinoid receptors in periphery is enough to completely bring the exercise performance of these mice down to the antibiotic level control, basically suggesting that the fatty acid amides and all these microbes can no longer provide a beneficial effect by stimulating the sensory neurons in the periphery. And this, again, on the level of the central nervous system was regulated by inability of the sensory nervous system to suppress the MAO-A, which then, of course, led to accumulation of enzyme that is degrading the dopamine, that then basically degrades it, and there is not enough dopamine to boost our motivation to basically perform well. So one of the very last piece of data was actually kind of like try to tie this all together. And in order to do that, we basically used only antibiotics drinking mice, and half of them were fed with the diet. Just as we saw in all the experiments before, their performance significantly increased by basically stimulating this peripheral nervous system. But then when we started injecting them with the M6545, which blocks basically the beneficial effect that these fatty acid amides can kind of like produce onto the nervous system, this is completely ablated, and basically there is no beneficial effect of the supplementation anymore in the mice that are lacking the microbiome. So when we basically just summarize what I've just showed you, is that we don't need just some microbes, but we need relatively particular microbes. Like here I showed you an example of Eubacterium rectale or Coprococcus entactus, that are then producing these very specific metabolites like fatty acid amides and oleoethanolamine or many others, that can then relatively specifically bind to cannabinoid receptor 1 in the sensory neurons, that then in the can go basically through the dorsal root ganglia to the spinal cord and suppress on the level of the VTA and nucleus accumbens the expression of the MAO-A, that then leads or results in the high levels of the dopamine that in the long term then increases our pleasure seeking and pleasure feeling from the exercise, and it kind of makes us want to kind of do it again and again and again, which is then of course very important in long term for all the benefits that we can receive from the exercise that can modulate our physiology in a good way. So with this, I will end here. It was a really huge team effort. As you can imagine, it's very, very complex. So I would want to thank here everyone from Christoph's lab, and then especially Nick's Berkeley lab, who are like a great specialist on all these brain recording and imaging, which wouldn't be possible without them. And then I would like to thank all of you for your attention. And if you have any questions, I would be happy to answer them.